So we'll discuss today about mechanism of allograft rejection. In the last video we learned about what different types of grafts are, how grafts are introduced into the body, and about auto and iso, allo and xenografts. So as you know, as we already talked, that most of the grafts that are done are allografts. And there is a good amount of chance that these allografts get rejected, and we'll discuss about the mechanism of allograft rejection. The allograft rejection actually can occur by cell-mediated immunity, by humoral immunity, and NK cells can also play an important role in them. So we'll discuss this one by one. So in terms of cell-mediated immunity, recipients T-cell mediated cellular immune response against allogeneic antigens on grafts. That means those antigens on grafts which are different, most commonly the MHC molecules, they are responsible for this kind of development of rejection of the graft. So the molecular mechanisms that are actually responsible for this are the T cells of the recipient recognize the allogeneic MHC molecules, that is the molecules of MHC and molecules which are different in the host and the recipient, between the donor and the recipient. Then there are these number of 10 to 4 to 10 to 5 to 10 to 4 of specific T cells that recognize conventional antigens, and 1 to 10% of T cells recognize allogeneic molecules. They have the capacity to recognize them. So many T cells can recognize allogeneic MHC molecules. So they can recognize allogeneic MHC molecules and then can mount a response against them. So recipient T cells recognize the allogeneic MHC molecule. There can be direct recognition and indirect recognition. We'll see what this is. Direct recognition of an intact allogeneic MHC molecule displayed by donor APC in the graft. That means the allogeneic MHC molecule present in the donor tissue can be actually recognized by the host T cell receptor directly, and sometimes cross recognition can occur. That means an allogeneic MHC molecule with a bound peptide can mimic the determinant formed by a self MHC molecule plus foreign peptide. A cross reaction of a normal TCR which was selected to recognize a self MHC molecule plus foreign peptide with an allogeneic MHC molecule plus peptide. So this type of recognition can also occur. So if you see, then you have a foreign peptide and self MHC binding. That means a foreign peptide which is presented by the antigen presenting cell, and then it is recognized, and peptide dominant binding, antigen presenting cell, and then MHC dominant binding. So in this case what happens is that self MHC molecule in the first case with foreign peptide can be recognized by the T cell receptor. Then you have peptide dominant binding, where you have the peptide, and then you have non-self MHC2 molecule, that means from the donor it can be recognized, and then MHC dominant binding, that means MHC molecules can bind to the T cell receptor of the if you see, the T cell receptor in here is from the recipient, whereas the antigen presenting cell, the MHC molecule, is from the host. So that is what is meant by these type of recognitions. These are the different types of recognition that can occur and cause the rejection of a graft. Now, passenger leukocytes. So donor APCs that exist in grafts such as dendritic cells and macrophages. So sometimes the antigen presenting cells are present in the donor tissue, and they can be directly recognized by the recipient T cell receptors. That is actually the cause for early phase of acute rejection, and this is actually fast and strong rejection which is seen. Okay, so many T cells, they have, as we already discussed, that they have the ability to recognize allogeneic MHC molecules. Allogeneic MHC molecules have different residues than the host, than the recipient MHC molecules. So allogeneic MHC molecules can bind to different peptides, and all allogeneic MHC molecules on donor APCs can be epitopes recognized by TCR. So that actually forms the basis of rejection, different basis of rejection. That means sometimes the allogenic MHC molecules can be recognized by T cell receptors as foreign, and then a response can be generated against them. And allogenic molecules can bind different peptides, and they can also be recognized as foreign, and then the rejection reactions can happen. Indirect recognition. The indirect recognition means the usual route is followed. If you see in the direct recognition, there was no internalization of foreign antigen by the antigen presenting cells, and then those molecules being degraded into small peptides and then presented to the through the antigen presenting pathway. That doesn't occur. But in indirect recognition, this is the mechanism that is followed. That means uptake and presentation of allogenic donor MHC molecules by recipient APC in normal way. That means the normal antigen presenting pathway in the host in the recipient actually operates in this. The from the antigens from the donor are processed through the antigen processing pathway, and then they are presented to the recipient T cell receptors and other molecules that can generate a response. So recognition by T cells like conventional foreign antigens. This is the process, normal process. Whereas in direct recognition, the antigen doesn't go through the antigen processing pathway. So if you see, this is the representation, the direct allorecognition, allogenic antigen presenting cells in the graft. Then you have allogenic MHC, alloreactive T cell, and a direct kind of a interaction between the antigen presenting cell of the donor is occurring with the recipient alloreactive T cell, and unprocessed antigens are recognized, and then a response is generated. Whereas in case of indirect alloantigen representation, you have the same process that is recruited as in the case of foreign antigens. This is again a type of representation of the same. So the indirect recognition is generally seen is slow and weak, and it occurs in the late phase of acute rejection and chronic rejection, and coordinated function with direct recognition rejection can also occur. So this is the difference between allogenic MHC molecule in between direct and indirect recognition. So you can see what exactly is the difference and what type of responses are generated. This we have already discussed in the previously. Now activated CD4 T cells by direct and indirect recognition, they can stimulate the cytokine secretion, macrophage activation and recruitment, and activated CD8 plus T cells can by direct recognition can kill the graft cells directly, which is the normal procedure for both of these TH and TC cells. Activated CD8 cells by indirect recognition cannot kill the graft cells directly. That is sure you know that. This is how this happens, sensitization, and then you have the effector response. So donor antigen presenting cell class 2 class 1 is being represented. Then we have CD8 plus that is your cytotoxic cell, and then you have helper cell, and then they can mount a response and effector mechanism takes place. So if you see, this is how it happens. CD4 TH1, and then you have the proliferation of these cells, CD8 plus pre-CTL, and then you have proliferation because of the binding, and then you have CTLs, and which can actually cause the direct killing of the cells. Graft cells. Now humoral immunity, important role, it plays an important role in hyperacute rejection. Preformed antibodies are present, and complement activation can take place. Antibody dependent cell mediated cytotoxicity can take place, that can also contribute towards, and then opsonization can also happen. It can be controlled by blocking antibodies. Natural killer cells, they can't recognize the allogenic MHCs on the graft. As you know, they lack specific receptors, and then cytokine secreted by activated TH cells can promote natural killer cell activation, and they can be recruited. So these are the mechanism of graft of rejection, the different type of cytokines that are secreted, and then you have reactive oxygens, the nitric oxide, inflammation. Different types of responses can be generated by virtue of different cytokine profiles that are secreted by the Th cells. So this is, you have an inflammatory response dependent of first is the inflammatory response, then you have a predominantly antibody response which can cause ADCC, and then you have the TC cell recruitment which can actually lead to rejection. I will talk about types of graft rejection. The types of graft rejection are hyperacute, acute, and chronic rejection. So we'll discuss this in a subsequent lecture, and then we'll finish up with the graft, the transplantation immunology, or predominantly. I have already sent to you the the parts of the book which you have to read and get acquainted with this. So if in case there are any queries, you can email me or you can send a message on WhatsApp as is feasible to you. Thank you.